do some techniques under chapter 2, techniques used in the study of cell biology. Last class, I discussed with you 3 techniques on phase contrast microscopy, types of phase contrast microscopy, application of phase contrast microscopy, limitation of phase contrast microscopy. Next, confocal microscopy, therefore types of confocal microscopy and use of confocal microscopy. Then I also discuss phase contrast microscopy and confocal microscopy differentiation. Then fluorescence microscopy, parts of fluorescence microscopy, advantages and limitation of fluorescence microscopy. Today, I will start a new technique, atomic force microscopy. Atomic force microscopy or scanning force microscopy is a very high resolution type of scanning probe microscopy with demonstrated resolution on the order of fractions of a nanometer, more than 1000 times better than the optical fraction limit. Thus, in this technique, we used atomic force for the detection and scanning of any object. Advantages of first commercially available atomic force microscopy of introduced 1989. The atomic force microscopy is one of the foremost tools for imaging, measuring and manipulating matter at the nanoscale. Just we will measure the object at the nanoscale level in this technique. Advantages. The atomic force microscopy has been applied to problems in a wide range of disciplines of the natural sciences, including solid-state physics, semiconductor science, technology, molecular engineering, polymer chemistry, physics, surface chemistry, molecular biology, cell biology, and medicine. In molecular biology, atomic force microscopy can be used to study the structure and mechanical properties of the protein complex and assemblies. Finally, the atomic force microscopy can operate in a number of modes depending on the application. In general, possible imaging modes are divided into static modes and a variety of non the atomic force microscopy last application is the atomic force microscopy can be operated in a number of modes. We will apply this technique in different modes depending on the application. In general, possible imaging modes are divided into static or contact modes and a variety of dynamic modes where the cantilever is vibrated or oscillated at a given frequency. Different modes. We want to use this technique for different modes for detection of the different levels of particles and different size of particles in a high resolution. Some disadvantages also found in these techniques. The disadvantages of atomic force microscopy compared with the scanning electron microscopy is a single scan image size. When it is differentiated with electron microscopy, then it is single scan image size. It shows a single scan image size. Another disadvantage is atomic force microscopy images can also be affected by non-linearity, heterosis, and creep of the piezoelectric material and cross-link between XYZ axis and may require software enhancement, and filtering. When we want to show the image with the graphical presentation, then some limitations shows in these methods. Last, due to the nature of the atomic force microscopic probes, they cannot normally measure steep walls or overhang. Sometimes this problem may occur. Next technique, video enhanced microscopy. Video enhanced microscopy combined with the use of a computer frame, memory extended, consider ably the useful range of our video enhanced contrast method for polarizing double beam interference and differential interference contrast microscopy. Just in this technique, used different types of interference to see the image. And in this image, after the visualization or scanning of the object, we want to found this video, we want to found this image as a video clip. Such video enhanced microscopy has led to renaissance in the application of the light microscopy for the studying of living cells in two important areas. Just two important areas: the intensification of faint fluorescence image permitting observation of fluorescently labeled cells under condition of very low illuminating intensity. Number B, the enhancement of extremely low contrast images generated by minute cellular structure so that they may be clearly seen and their normal intercellular movement recorded. Next, features of some video enhanced microscope. Features: the combination of novel optical microscopic technique with advanced video and digital imaging technology now permits dynamic improvement in the quality. In this technique, advanced video and digital imaging processing technology used to perform the successful operation. To video enhanced microscopy required the combination of a versatile high quality research microbes with an analog and digital real-time image processor. Last, application of video enhanced processor has recently led to major discoveries concerning the functioning of the living cell. Next technique, electron microscopy. Electron microscopy is a technique for obtaining high resolution images of biological and non-biological specimens. In this technique, we used for the find out of high resolution images of biological and non-biological specimen. It is used in biomedical research to investigate the detailed structure of tissues, cells, organelles, macromolecular complexes. Some history found for the invention and name coined the electron microscopy. The first prototype of electron microscopy was put together 1931 by physicist Ernest Rutherford and the electrical engineer Max Connell. For his work, Ernest Rutherford was awarded the Nobel Prize 1986 for the invention and full description of the electron microscopy. We found 4 variants of electron microscopy: immunoelectron microscopy, scanning electron microscopy, transmission electron microscopy, cryo-electron microscopy. Electron microscopy have 4 variants for the observation of different particles in different areas. Easily. Application of electron microscopy. Electron microscopy are used to investigate the ultrastructure of a wide range of biological and inorganic specimens, including microorganisms, cells, large molecules, biopsy samples, metals, and crystals. Last, modern electron microscopy produce electron micrographs using specialized digital cameras and frame grabbers to capture the images. You just read 2 or 3 applications as your easy. Disadvantage: electron microscopy are expensive to build and maintain. Just expensive, highly expensive, cost very high. But when we installed a electron microscopy, we want to use for a long time without any hesitation. Next, another limitation: samples of hydrated materials, including almost all biological samples, have to be prepared in various ways to stabilize them, reduce their thickness, and increases their electron-optical contrast scanning. Sometimes the thickness of the sample reduced in this technique and also increase their electron-optical contrast or staining. The staining level is very high in this technique. You also read 2 or 3 disadvantages. Next, immunoelectron microscopy. Immunoelectron microscopy is a method of performing an immunohistochemical method using an optical microscopy in an electron microscopy, and is called various names such as immunocytochemistry or immunomicroscopy, immunohistochemistry. Just in this technique, we use cytochemistry, immunocytochemistry in the electron microscopy as another technique for the detection of more samples in different fields. Steps of the protocol of immunoelectron microscopy. Steps: first, we washing the sample, fixation of sample. Next, again washing. Therefore, primary antibody incubation. Next, again washing. Secondary antibody incubation, washing, fixation. Fixation 2.5%, washing, PBS, RT, 15 minutes, 3 times. Osmication 1%, 4 degrees Celsius, 1 hour. 0 will be degree, not 0. 1 hour. Dehydration, drying, carbon coating. Last, observation. Immunoelectron microscopy, steps of protocol. Totally 14 steps. Scanning electron microscopy. Scanning electron microscopy is a type of electron microscopy that produces image of the sample by scanning of surface with a focused beam of electrons. We will scan the surface with a focused beam of electron. This technique is called scanning electron microscopy. The electrons interact with the atom in the sample, produced various signals, and contain information about the surface tomography and composition of the sample. Advantages of scanning electron microscopy. Advantages: resolution. In this microscopy, the sample's image will be provided by high resolution, therefore traceable standard for magnification. Magnification also will get the magnification level also high. Chemical analysis. Scanning electron microscopy with EDS provides quality chemical and elemental analysis, standardized quality quantification analysis, X-ray line scan, and mapping. This data can be used to examine product defect, identify and the elemental composition of foreign material, assess the thickness of the coating, and determine gain and the particle size. These 3 options is a advantages of scanning electron microscopy, and 3 disadvantages also occur during the scanning electron microscopy operation on vacuum environment. In most cases, scanning electron microscopy must be solid and vacuum part compatible. However, higher pressure can be used for imaging for vacuum sensitive sample. Just some samples are sensitive under vacuum. In this technique, needs high pressure for the scanning of the some samples under vacuum environment. Articrafts are possible. Samples that are strong insulator must be coated usually with gold or carbon before testing. It is another disadvantages. Others disadvantages on possible, others parties application, not disadvantages. You will read this area as application in scanning electron microscopy. I will replace this point under application and send you a new sheet. So disadvantages only to vacuum environment and artifacts are possible. Just two options are disadvantages. Transmission of electron microscopy. Transmission electron microscopy. Transmission electron microscopy is a microscopy technique in which a beam of electrons is transmitted through a specimen to form an image. Transmission electron, just transmission of electron through a specimen to form an image. This technique is called transmission electron microscopy. The specimen is most often an ultra thin section less than 100 nanometer thick or a suspension on a grid. An image is formed from the interaction of the electrons with the sample as the beam is transmitted to the specimen. This technique is called transmission electron microscopy. Components of transmission electron microscopy, you just read only points, no need detail. First, vacuum system. Vacuum system is the first component of transmission electron microscopy. Second, specimen states. Third, electron gun. Fourth, electron lens. Last, aperture. 5 components is generally required for the finding of a transmission electron microscopy system. Next, properties of transmission electron microscopy. Properties of transmission electron microscopy. Transmission electron microscopes are capable of imaging at a significant higher resolution than light microscope. It is very helpful, higher than light microscope, owing the smaller the Golgi wavelength of electron. Last, transmission electron microscopy finds application in cancer research, virology, material sciences, as well as pollution, nanotechnology, semiconductor research, but also in other fields such as paleontology and palynology. We Bengali need to Bengali called Protnotattva. Some paleontological object identification and also determine the year of this particle. This type of microscope is generally used for the scanning and imaging. Limitation of transmission electron microscopy. Limitation: many materials require expensive sample preparation. To produce a sample thin enough to electron transparent, which makes transparency in electron microscopy analysis is relatively time consuming process with a low throughput of samples. Just two limitations in this point: require expensive sample preparation on, and another limitation, time consuming. Sorry for disconnecting. Electricity was lost and internet connection was disconnected. Next, limitation, another limitation. First, two limitation in this first point: sample preparation, expensive sample preparation, require expensive sample preparation. Due to sample preparation need, highly expensive, another limitation, relatively time consuming from another techniques. Point 3, there is potential when the sample may be damaged by electron beam, particularly in the case of biological material. When we use biological material, sometimes it will be damaged. So need careful handling and precaution in this technique. Transmission electron microscopy has three essential systems. Three essential systems needed for the operation of transmission electron microscopy: on an electron gun which produces the electron beam, and the condenser system which focuses the beam onto the object. Onto the object, the electron producing system consisting of the object lens, movable specimen stages, and intermediate and projector lenses which focus the electrons passing through the specimen to form a real highly magnified image. Last, the image recording system which converts the electron imaging into some form perceptible into human eye. The image recording system using consists of a fluorescent skin for viewing and focusing the imaging and a digital camera for permanent records. In addition, a vacuum system consisting of pumps and their associated gauzes and valves and power supplies are required. Those are the three essential systems in this technique. Last technique, cryo-electron microscopy, is an electron microscopy technique applied on the samples cooled or cryogenic temperature and embedded in an environment of vitreous water. An aqueous sample solution is applied to a grid mesh and pans frozen in liquid ethane or a mixture of liquid ethane or propane. Cryo, just we know cryo means frost, highly cooled, very low temperature, minus temperature. And also this technique used liquid ethane and propane, vitreous water to maintain the temperature to operate the cryo-electron microscopy. Cryo-electron microscopy have some characteristics. Cryo-electron microscopy is gaining popularity in structural biology. Cryo-electron microscopy day by day gaining the popularity in structural biology to the visualization of different samples. Cryo-electron microscopy is form a cryogenic electron microscopy, more especially a type of transmission electron microscopy where the sample is studied and a cryogenic temperature. The main advantages of cryogenic microscopy is freezing samples, minimizes the radiation damage that samples can suffer as a result of electron irradiation. Next features, the technique allows scientists to study the objects that, because of their size, complexity, or sheer awkwardness, would be virtually impossible to scrutinize with the other technique. It's quick and flexible. This method is quick and flexible, and when used the tandem with other techniques, cryo-electron microscopy is supremely powerful tool with the other techniques. Last, an application of cryo-electron microscopy is cryo-electron tomography, where a 3D reconstruction of the samples is created from the titled 2D images So in this technique, if when an images are 3D reconstructed, if we want, we will transfer to 2D images version. Thank you.